We get rolling, and I don't want to cut into anybody's time. I just want to say thank you, everybody, for coming out. Thank you to all our speakers and everyone who submitted an abstract for this. I'm pretty excited for it. I think it'll be a really awesome symposium today, and I really hope that this can help foster some discussion across some different fields. And so our first speaker will be Emily Kopania, talking about the evolution of high visual acuity specializations in mammals.
Hello, everyone. My name is Emily Kopania, and I am a postdoc at the University of Pittsburgh. Is the microphone working? Okay, thank you. I'd like to start by thanking the organizers for putting together this symposium. Across mammals, we see a wide range of variation for many ocular or eye-related traits. Some of these are morphological traits that we can easily observe, such as the relative size of the eyes and the angle of the eyes in the head. We also see variation in mammals for many cellular level ocular traits, such as the density of cells in the retina. This variation is often thought to reflect differences in the visual environment in which these species live and forage. However, vision is a complex sensory system that requires the successful integration of these morphological and cellular level traits, which raises the question, what is the relationship between the evolution of morphological and cellular ocular traits? One morphological trait that varies widely across mammals is orbit convergence angle, or the angle of the eyes relative to the head. Some mammals, such as this rabbit, have side-facing eyes corresponding to low orbit convergence, and this is thought to give them a larger field of view or sort of more panoramic vision. Other species have more front-facing eyes, corresponding to a high orbit convergence angle, and while this probably reduces their overall field of view, it leads to more overlap between the field of view of the left eye and the right eye, and this is thought to facilitate better depth perception and higher visual acuity in front. So many of you have probably heard this popular idea that predators tend to have front-facing eyes, and prey species, or species that tend to have higher predation rates, are more likely to have side-facing eyes. However, most studies that have investigated this in mammals have done so within specific clades, such as primates, or have not properly accounted for shared evolutionary history among these closely related species that could influence various traits. So I set out to address this question. Are front-facing eyes associated with predation across eutherian mammals if we correctly control for phylogenetic relationships? To do this, I measured orbit convergence from skull specimens at the Carnegie Museum of Natural History. We measured orbit convergence as the angle between the sagittal plane, or the plane through the center of the skull, and the orbital plane, or the plane through the eye socket, shown here in yellowish green. And to do this, we had the machine shop at the University of Pittsburgh build us a custom angle measuring tool, following established methods from previous papers. So we measured specimens from about 30 species, and combined this with orbit convergence data from the literature, and overlaid these data on a mammal phylogeny. So in this image, the warmer, redder colors correspond to species with higher angles or more forward-facing eyes, and cooler colors respond to more side-facing eyes. So we estimated Pagel's lambda to be 0.78, or significantly different from 0, suggesting that we have high phylogenetic signal. So in other words, shared evolutionary history has a big influence on patterns of orbit convergence that we see across mammals. I then tested if orbit convergence is associated with predation broadly across mammals, and was quite surprised to find that we did not get a significant result here. So looking at these uncorrected points, there is a trend towards, on average, higher orbit convergence in predators, consistent with previous studies. However, when we apply a phylogenetic generalized least squares test, we do not find statistical support for a difference here. And similarly, I used herbivory as a proxy for likely prey species, and again did not see a significant difference in orbit convergence between these two groups. So this is one morphological trait in mammals. As I said earlier, we also see variation in many cellular traits, and one example is retinal ganglion cell density. So this image here represents a retina that has been cut in several places so that it can be flat mounted on a slide, and it's oriented such as the dorsal side is on top, ventral on the bottom. And for retinas and eyes, we mark the nasal and temporal ends. So think of nasal as being closer to your nose, and temporal as the other side, it's closer to the temple of the skull. Many studies have made these flat mounts for different species and stained for retinal ganglion cells. These are important because they send the visual signal directly to the brain, and their relative density is thought to correspond to visual acuity, or how sharply we can see an image. So across many species, there's variation across the retina in ganglion cell density, with some low-density regions and some very high-density regions, which presumably correspond to the parts of the retina that have the highest visual acuity. So studies will make these retinal topography maps that indicate the density of ganglion cells across the whole retina. They look like this. And a couple common patterns occur that we refer to as retinal specializations. So one common pattern is called the area centralis, and this is where density is really high in one region of the retina and then decreases as you move radially outwards towards the periphery. Another common pattern is the horizontal streak, where density is high across this temporal nasal axis in the center of the retina and then decreases as you move towards the dorsal or ventral sides of the retina. And during this talk, I'm going to use these cartoon images to indicate either the area centralis or the horizontal streak. Many have proposed hypotheses for ecological conditions that could potentially select for these different types of retinal specializations. One prediction is that the area centralis will be common in predators, and another is that the horizontal streak will be associated with species in horizon-dominated environments. So to test these predictions, I gathered data from the literature on the presence of different retinal specializations across mammals and overlaid these on a phylogeny, and then combine these with existing ecological data. So in this image, the purple boxes are species with an area centralis, and the yellow boxes are species that are considered predators. And when I tested for a relationship here, I did not find a significant association. However, I did find a significant relationship between the horizontal streak and ground foraging. So ground foragers are the ones with pink boxes, and I'm using this as a proxy for foraging in a horizon-dominated environment, in contrast to a complex three-dimensional environment, such as marine or arboreal environments. And species with a horizontal streak are shown with orange boxes. So we do find a significant relationship here between the horizontal streak and ground foraging. So there's mixed evidence for some of these predictions for environments that these retinal specializations will be associated with. And as I mentioned earlier, there's likely some relationships between these morphological traits and cellular level traits to integrate these different levels of eye morphology. So one prediction is that the horizontal streak will be associated with side-facing eyes because they're both thought to facilitate higher acuity in a horizontal plane and more panoramic vision. When I tested this, I found that the horizontal streak is significantly associated with more side-facing eyes. So shown here in orange, these are species with a horizontal streak, and they tend to have significantly lower orbit convergence values compared to species with an area centralis. There are also predictions related to orbit convergence and the position of the area centralis. So despite its name, the area centralis is not always actually in the center, and in many mammals, it's actually shifted towards the temporal side of the retina, shown here in sort of the red splotches. So one prediction is that this is to facilitate higher visual acuity in species that have more side-facing eyes, because this area centralis will project forwards if it's shifted temporally. So to illustrate that, if we imagine species with low orbit convergence and an area centralis located in the center, the regions of highest visual acuity, shown in these darker cones, will project sideways. However, if the species with the same low orbit convergence has an area centralis shifted towards the temporal side, then they'll project forward because of the way the eye is curved and lead to higher visual acuity in front despite these side-facing eyes. So this has led to the prediction that there should be a negative correlation between orbit convergence angle and the temporal shift of the position of the area centralis. And some species do support this idea. So for example, the sheep has very side-facing eyes, and an area centralis almost entirely on the temporal side of the retina, shown here in this dark reddish-orange color. So a few species back up this idea, but this hasn't been tested quantitatively across a broad sample of mammals. When I did this, I found evidence for a strong negative correlation between the temporal shift of the area centralis and orbit convergence, supporting this prediction. So in the bottom lower left there, those will be species with more front-facing eyes and a centrally located area centralis. And then in the upper left-hand corner, that will be species with very side-facing eyes and an area centralis shifted almost all the way to the temporal side of the retina. So we find strong evidence for this relationship, and this p-value is based on a PGLS analysis. So some concluding thoughts on this. I think that orbit convergence may be under strong constraints due to other aspects of skull morphology. So if we look at this rabbit and this leopard skull, one has very side-facing and one has very forward-facing eyes. But there are also many differences between these two species in terms of their skull morphology that could indirectly influence their orbit convergence angle. In contrast, retinal specializations may experience fewer constraints and therefore be more free to evolve in response to both ecological pressures and also functional interactions with other ocular traits. And indeed, the strongest trend we saw was this relationship between orbit convergence angles and retinal specializations. So perhaps they are evolving in response to potential constraints imposed on other eye traits. So with that, I would like to thank my lab mates, especially my PI Nathan, for their support on this project, as well as the museum and university resources that made this work possible. I'd also like to acknowledge several colleagues I've had helpful discussions with and my funding sources. And I think there's time, so I will take questions. Yes.
Did you look at time of day of activity of the area centralis and go away on nocturnal species?
Yeah, we did not look at that for the retinal specializations. We did look for the orbit convergence angles and did not find any significant relationships with that either. Yeah. Okay. Oh, yes.
So when you're thinking about constraints that skulls might impose, is that things related to like bite force or
Yeah.
imagining broadly enough?
Yeah, sort of bite force. Some studies have thought about kind of the rostrum, so actually the width and the length of the rostrum. So there's some evidence within, I think, marsupials that could affect things, but I'm not sure about more broadly across mammals. Yeah. Yes.
Is there any sort of potential evidence that suggests why a certain side-facing, or I guess you kind of explained it, but like the evolutionary history of side-facing eyes that don't have that horizontal streak?
Yeah. So potentially, I guess, thinking about the position of the area centralis, like I was thinking about, I guess another thing that I sort of didn't have time to get into is some species can have multiple specializations. So they can have like an area centralis within a horizontal streak, and sort of to simplify the analysis, I focused on ones that had only a horizontal streak, but some of them might have a more complicated specialization, kind of combining those patterns. Yes?
Primates are an interesting group in this because they're often both predators and prey. They're using binocular vision for fine motor control tasks as well. Is there
Right.
anything unique or different about primate ocular adaptations and some of the measures that you've taken?
Yeah, so primates do sort of stand out in their orbit convergence values, even relative to like carnivores, for example. They have very high orbit convergence. And then I didn't get into the details on this, but primates actually have a fovea, which is like an area centralis with a pit in the center. So they do have very unique eye morphologies. But yeah, it's because they're sort of the only group within mammals that have these, it's difficult to like take these tests, right, and compare those different ecologies. Thank you.
So, hi everyone. Thank you for coming. Today I'm going to be talking about differences in sensory weighting as well as visual acuity into ecologically divergent heliconius butterflies. So, as you all know, divergent selection between populations can lead to local adaptation. If you think of populations living in different environmental conditions, this can lead to local adaptation, and this also happens in their sensory systems, such as their visual system or olfactory system, which can adapt to their local conditions, and this can lead to differences also in behaviors. So for example, you can think that mate choice might change between different populations that have different sensory systems, but also differences in other behaviors such as foraging ability or predator avoidance could also change. For this project, I was looking at Heliconius sydno and Heliconius melpomene. These are two closely related species that live in sympatry. However, they're separated across an ecological gradient from open forest, where we find Heliconius melpomene, and then with decreasing light intensity as we go through the more close part of the forest, we find Heliconius sydno. Interestingly, these two species also differ in neuronal investment, where, as you see here in the brain of the butterflies, the blue parts of the brain are associated with visual processing, and these are larger in Heliconius sydno than Heliconius melpomene, and this is likely to do because of the different light environments in which they live. It's important also to mention that these differences are not only seen on wild individuals, but also there are heritable differences, and individual and hybrids of these two species are intermediate. In addition to this, there's also differences in eye morphology. So Heliconius sydno have larger eyes than Heliconius melpomene, but also they have more facets, which are the individual light sensing units in insect eyes. So for my project, I wanted to see what the behavioral implications could be of this increased visual investment in Heliconius sydno. And first of all, we wanted to see whether there are differences in sensory weighting, because we thought that likely, since Heliconius sydno has a larger investment in visual behaviors, might be more strongly weighted than olfaction in comparison to Heliconius melpomene. So for this, what we did is we trained groups of butterflies of the two species to associate food reward, which in this case were blue feeders with rose smell as a positive reward, or red feeders with a lavender smell as a negative color and odor combination. And we did this over the course of a few days to first condition the butterflies, and then we tested them to see whether there are differences in sensory weighting. So I'm going to show you this graph, and I'm going to walk with you a little bit so that you understand. On the y-axis, you see the proportion of choices towards blue feeders, and then on the x-axis, you see the different color and odor combinations. So for example, on the naive day and on the trained day, you see that the combinations are blue with rose or red with lavender, whereas in the conflict day, which is the main test that we're trying to see, we've switched the positively rewarded color with the negatively rewarded smell, so blue with lavender or red with rose. And then on the 7th day, we only presented the butterflies with visual cues in the absence of odor cues. And importantly, throughout all these tests, so we trained the butterflies always after we've tested them, but during the test, there's only water, so what we're seeing is already predetermined behaviors that the butterflies had learned. So first of all, when we look at the naive preference, we see that both species mostly prefer the red feeders. This is because most Heliconius butterflies have an innate preference for red feeders. However, if we condition them for a certain number of days, we can also train them to like this particular combination of blue feeders with rose smell. And what's really interesting is that if we switch the positively rewarded smell with a negatively rewarded odor and vice versa, we see this strong behavioral difference between the two species. So Heliconius sydno still mostly relied on visual cues, so that's why most of their choices are towards blue feeders, whereas Heliconius melpomene mostly relied on olfaction in comparison. And then when we presented the butterflies with colors only, we see that both butterflies are using visual cues, given that both prefer the blue feeders. So this is showing us that first, both species are using vision and olfaction in their foraging choices. However, the relative weights given to one or the other differ, with the species that has a larger investment in vision mostly relying on vision in comparison to Heliconius melpomene, which mostly relies on olfaction. And then another behavioral implication that we wanted to see is whether there are also differences in visual acuity, which is the ability to perceive a static spatial detail between these two butterflies. However, we cannot ask the butterflies, give them an eye chart and ask them whether they can see or not. So what we did is we did this optomotor assay, which I'll show you here. I promise you the butterfly is moving. We've done this many times. So we have alternating black and white stripes, and as you can see, when the drum moves, the butterfly moves its head. And this is a needed response that animals have when they can perceive motion happening, but as the stripes get thinner and thinner, at some point the butterflies are not able to perceive more this difference in motion, and they won't move their head. So we've tested this with many, many butterflies of both species as well as their hybrids, and what we saw is that here you can see on the y-axis the visual acuity. In all the plots that you'll see, Heliconius cynodon is highlighted in blue, and Heliconius melpomene is highlighted in orange. The species with higher visual acuity is also the species that has larger investment in eyes. But interestingly, there's also sex differences, with males having higher visual acuity than females. And we think this is likely to do because of their differences in biology, where males spend most of their time trying to find suitable mates, whereas females spend most of their adult lives finding plants to oviposit. But what we also found that was quite interesting is that we know that the brains of these butterflies keep developing after the butterflies eclose. So we wanted to see whether there are differences in visual acuity related with age, and we see that visual acuity changes in the adult butterflies, and it reaches its maximum at around 6 days, which is where males start courting females and they reach adulthood. So as I told you before, the fact that the butterflies that Cydno has higher visual acuity than Heliconius melpomene kind of made sense, given that we know that they have more facets and larger eyes. But what's quite interesting is that there are no age-related differences in the eye size or facet number between the different ages, which means that these differences in visual acuity that we see with age might be explained with something else. So now what I'm trying to do is so we fixed brains of butterflies of different age stages, and we're trying to test whether, first of all, confirm that there are species differences and sex differences, but also we want to see whether there are indeed differences in the brain and the visual investment that are changing with age as the butterflies develop. And then the last thing that we want to do is we're also harnessing the ability to make crosses between these species, and also back crosses to separate out the different components of visual acuity. So in that way we can separate, for example, differences in eye size or corneal area, but also differences in neural investment. And as you can see here, we've tested already about 100 back crosses in each direction, and we see, as expected, back crosses to Cydno have higher visual acuity than back crosses to Heliconius melpomene, and then the hybrids are intermediate. And with that, I'd like to thank you so much. This is just a quick summary, a take-home message, if you will, of what I've been talking about. So we know that these species live in different light environments, and they have invested differently, not only on the visual periphery, not only on the eyes, but also on the brain, and this has led to behavioral changes, both in sensory weighting, but also in visual acuity. And with that, I'd like to thank everyone here, and I'd like to thank everyone in Colombia, especially Danny and Geraldine, that helped me a lot with the projects, and everyone involved in this project. And thank you so much. I think we might still have a little bit of time, so if you have any questions. Yes.
Wonderful talk, thank you. Which areas of the brain are you going to use for visual signal?
So we're going to look at the visual neuropils, which are the outer volumes of the brain. So we're gonna test differences in all the there's 4 or 5 neuropils that are related with vision in these butterflies. So we're trying to see whether there are differences also with age. Yes.
Thank you very much for this talk. So you showed the differences between the 2 species. Just focusing on one species, would you expect if the populations are exposed to different light conditions, would they also be showing this species differences like they did in population?
That might well be the case. So we know that there are slight differences in populations, like across different areas, but in general, the patterns are consistent with Heliconius cygno having much larger eyes than Heliconius melpomene. But for example, I know that so these species that we tested here, these are populations from Colombia, and their eye size was a little bit smaller than some species that we tested in Panama. So there might be like small differences in the populations, but I think this is mostly important across species. So then, thank you very much.
All right, I'm going to go ahead and get started. My name is Maddie. I'm from University of South Carolina. Today I'm going to be talking about visual enhancement within tidal creeks. So visual contrast, this is necessary for animals to see any object against its background. It can be based on different principles of light. So presence or absence, you can think of black and white as seen in the top picture. We have color contrast, so this would be based on wavelength. And of course, polarization vision. So this is based on the angle of polarization or the degree of polarization. However, different light environments make it more difficult for animals to produce or enhance their contrast. So today what I'm going to be talking about are these tidal creeks. This is at North Inlet in South Carolina. At the Baruch Institute, what I really want to draw your attention to, there's a big variety of color within the estuary. So some is really green, some is really brown, different levels of turbidity. And so what we see are all these different obstacles to visual contrast. First of all, we have light scattering. You can think as you look through water, things look cloudy as light moves. This increases noise. We have light absorption. So I like to compare this to putting your tea into your cup and it turns brown. You can still see through it, but it's harder to tell colors apart, or like wearing rose-colored glasses. This decreases chromatic contrast. And then we have flicker. So if you've ever jumped in a pool and seen sort of that netting at the bottom, what this does is it reduces achromatic contrast. And then lastly, it's just really dynamic, right? These estuaries are constantly changing and shifting in brightness, different algal blooms. In the picture in the previous slide, what you saw was just a snapshot of what it might look like on one day. And even then, it's different throughout the estuary. So what this all leads to are all these factors reduce the contrast within a scene. So today what we're asking are, how do these 2 types of crustacean that have diverged very long ago handle the same obstacle of low contrast within their visual environment? So I'm going to be looking at these 2 crabs. We got bachelor number 1 up here. This is the green porcelain crab. These guys are anemomeran, so related to squat lobsters. I'd also like to point out they are invasive to the Atlantic flight, as well as they're filter feeders. So you can sort of see their little mouth parts. They do a little dance. It's pretty charming. And they have these sort of bulbous, reflecting superposition eyes. And secondly, we have our Atlantic mud crab, Panopius erbsti. These are true crabs, or brachyurans, so you can think blue crabs related to those. They are native, and they're actually ambush predators or foragers. And they have these apposition, sort of beautiful golden black eyes, but they are slightly bigger than the porcelain crabs. We're looking at these on scales of a few centimeters, so even the largest ones are about this big. So then how do animals enhance their contrast? I've kind of already introduced these. So we're going to be talking about polarization sensitivity, so looking at the angle of polarization or degree. You can see if you compare the left and the right picture, if the right picture has polarization contrast, you can notice all of those fish in that top right, that top right corner that you were missing without polarization sensitivity. Color vision, so if once we add sensitivity to wavelength, we get more detail. And then increased sensitivity, and what this really is increasing the saturation on your scene, so how much light is the individual capturing. So what we're hypothesizing is that they're using these three forms. So I'm going to test all of these. I'm going to run through them. But we're going to start right off with polarization sensitivity. The last talk did a great job introducing optimotor. We do this with the crabs. This is considered traditional or achromatic, so black and white stripes. You can see the crab following along. It changes direction. The crab thinks about it, and then he follows the directions still. And so they have a really great response. It's pretty easy to score. And so what we did was we tested their acuity, so comparing different widths of stripes. And then what we did was we created a polarized stimuli using polarized filter. And so what that was we arranged some of it perpendicularly to each other, these strips of polarized filter, and some parallel. And what this looks like in practice, to humans, we're not sensitive to polarized light. So what we would see is the top. This is our control and our 10 degree, our lackluster human eyes cannot see really the difference here. But once we look at this through a polarized filter, how crabs might see it, we can see the control is uniform, and then we have our polarized, which magically appears to these animals as they're sensitive. So what did we find? We found both of the crabs had about the same visual acuity. It's probably between 4 and 10 degrees is our estimate. This is pretty on par for benthic crustaceans of this size. So this was not very surprising. You can see, however, the porcelain crabs on the top, they don't like to move that much. They're pretty shy. So in general, they just don't respond as much to stimuli. What really surprised us, however, was that the porcelain crabs did not respond to the polarized stimuli, but the mud crabs actually did. So they followed the polarized stimuli about 14 seconds more than they would follow the achromatic control. So this is great evidence that they are sensitive to the angle of polarization of linearly polarized light. So what we wanted to know now is, can they use this in their environment to actually enhance contrast? We talked about these spectrally narrow environments due to like algal blooms. You know, they can be green, they can be red. And so what I tested was balancing LEDs to have the same light output and photon density under these 4 different colors. So we have a near UV, a blue, a green, and a red. And then we just did them with a control and the polarized experimental. And then we did it at 2 light levels. So we did it at a dimmer light level, I think it was about 10 to the 14th photons per centimeter squared per second, and then another about 40 times as bright. So what we found was, under dim conditions, they really didn't respond to the polarized more than the control. So this was a little disheartening at first, but then we repeated it, bumping up that brightness, and we did see that it was significant for blue and green and borderline on that 400 nanometer near UV we're considering. They do still follow polarized stimuli. So this indicates to us that these crabs might be using polarization contrast under spectrally narrow conditions to enhance their own contrast. So that's our answer here. Porcelain crabs, not so much. So now we're going to move on. I'm going to talk about color vision real quickly. What we did was we used electroretinography, or ERG, to measure eye responses to different stimuli. So what is this exactly? For those of you who are not familiar, you poke them in the eye with one electrode, you poke them in the body with another, and then right above that we have a light source. And what we can do is we can control the color of that light, the brightness of that light, and how fast that light flickers. We can run all of these different experiments. So this is what a visual response would look like. This is for different colors. So to the left would be a color they're more sensitive to, and the right less sensitive to. Similarly, if one color was brighter, they would have a larger response, versus dimmer, they would have a smaller response. And similarly, if you flash a light to see how quick their vision is, you can see it oscillating as their eye responds and sort of resets. Once they can't notice the flicker, it just becomes uniform. So what we did was we took the spectral response, we fit it to a curve, and then with the help of Dan Chappell from the Air Force Research Lab, we fit them to known visual pigments. And what we found was surprising was that the porcelain crabs were actually sensitive to 3 different visual pigments, it seems like. So they are actually trichromats. One around 480, one around 560, and this kind of surprising one around 375. You can even see the little lift in the data. We think there might be a 3rd photopigment in there that they are receptive to, and so maybe that's making up for the reason they are not sensitive for polarized light. Mud crabs, they have very typical crustacean blue-green sensitivity. This was not very surprising, about 490 and 555. So it does seem like they're using different color sensitivities to enhance their contrast. And then lastly, we looked at increased sensitivity. So once again, this is how much light is captured, how saturated does that scene appear to the viewer. And to do that, we continue to use ERG, but this time we looked at sensitivity, so dim to bright, as well as that flicker fusion, so how fast is the animal sampling the environment. If you're familiar with photography, it's sort of like shutter speed. So what I really want to draw your guys' attention to, this is relative response. We plan on fitting curves to this. What I want to point out is that porcelain crabs, even at the very dimmest light that we recorded, they had a larger relative response, so over 10% of a response to light. So what we expect is if we went even dimmer, the porcelain crabs would continue to be sensitive to a lower threshold than the mud crabs. So they seem to have this increased sensitivity, and then mud crabs, not so much. On the other hand, we ran how quick their vision is. What we were surprised to find was that porcelain crabs have estimated vision around 70 hertz. This is higher than what we thought we would find. Typically around 40, which is what the mud crabs have, is more standard for crustaceans around this size. And so what we think might be happening here is because mud crabs don't have really fast vision, they might just be expanding that sampling period to increase the amount of light they capture. So what we think this discrepancy in vision is probably due to is these porcelain crabs, they have super reflecting eyes, whereas the mud crabs have appositional eyes. So super reflecting eyes are really great at light capture. Appositional eyes, not so much. Further, the porcelain crab eyes anatomically probably do decrease polarization sensitivity due to the arrangement of its rhabdom. So lastly, I just want to leave you with a quick comparison, running you through everything I just really quickly went through. So just comparing porcelain crab versus mud crab. Porcelain crabs, not sensitive to angle of polarized light, probably due to eye structure, but mud crabs are, and under narrow spectrum light, provided there is enough light for them to create contrast. Color vision, it seems like porcelain crabs win this competition. They are more sensitive to a wider range of wavelengths. It might be trichromats, whereas mud crabs, less sensitive, only dichromatic. And then lastly, this increased sensitivity trade-off I just talked about. Porcelain crabs might be more sensitive with faster vision, whereas mud crabs are less sensitive with slower vision. And so what we're seeing are these two crabs that have very, very unique evolutionary histories that are both posing different solutions to enhancing contrast within the same environment. With that, I'd like to thank my advisor, Dan Spicer, as well as lab members, Dan Chappell, my committee, as well as all of the quote-unquote undercrabs who have helped along the way with this research, and I will take questions with a little bit of time I have left. Yeah.
So, assuming that the other senses of the two crabs are the same, ecologically speaking, would you say that it would make some evolutionary sense that the crab that is able to navigate its environment and find prey has more light sensitivity?
To some degree, yes. We think the porcelain crabs might have increased sensitivity because they're filter feeders. It's really important for them to position themselves properly to maximize their capture. What we really want to do is go out to those tidal creeks and take measurements to see what might be driving it more hard. The other thing is, like I said, the porcelain crabs are invasive. Their native environment tends to be slightly clearer water. Yeah.
How ecologically relevant do you think those
Yeah, probably. Light is just changing really fast in terms of brightness and whatnot. We're not exactly sure. Generally, faster vision is better for motion capture. So porcelain crabs, I didn't quite mention it, everything really loves to eat them. Their shells are really thin. Natives just love eating them. And so it might just be that they're more sensitive to movement to avoid predation. The mud crabs have much thicker carapaces and are much more aggressive. Anybody else? Yeah.
Just a basic question. Do these crabs have glass cells? And also among the cones that you're saying, which one is sensitive to the polarization?
The mud crab is sensitive to polarization. And what was your first question?
Which one? Like you have, like they have 2 or 3 cones, right?
Yeah, they have, the porcelain crabs are not sensitive, but they have more wavelength sensitivity, and the mud crabs are sensitive, but don't have as great color vision.
Like all 3 cone types are sensitive to polarization?
No, only the mud crabs are sensitive to polarization. Generally, all crustaceans are, though. Sorry, yeah, I'm out of time.
So, hello everyone, my name is Joséphine. I'm a second-year PhD student at the Museum of Natural History in Paris. And today I'm going to talk about the use of visual cues in species recognition during courtship in morphobutterflies. So color and color perception are really important in mate choice in many animal species, and this is especially the case in butterflies. You can see that they have a very diversified coloration and color pattern on their wings. We also know that diurnal butterflies have very good color vision, and quite a few studies have shown that they use visual cues in order to locate their mate in their environment or discriminate their mates from other butterflies living with them in the same environment. And this discrimination behaviors and capacities are very important and have likely evolved because of reproductive interference. So we consider that the hybridization between 2 different butterfly species is likely deleterious and costly in terms of energy. And so the butterfly showing good discrimination and visual discrimination in this case often have a better fitness than the other butterflies, which explains why visual discrimination is important in those species. So during my PhD, I've been studying the use of visual cues during courtship in morphobutterflies. So morphobutterflies are neotropical butterflies that can be found in Central and South America, and they are characterized by this very striking phenotype, which is called iridescence. So you can see here that morphos are mainly blue, but depending on the angle of illumination or observation, we can see different color variation on the dorsal side of their wings. And there are quite a few hypotheses trying to explain why this type of phenotype has evolved. And today I'm going to focus on trying to study the effect of sexual selection and the vision of iridescent cues during courtship in those morphos, because we have observed what looks like a color display behavior during courtship in Morpho-Eleanor in particular. And so I'm going to study the use of visual cues and the perception of visual cues in Morpho-Eleanor in particular. So here you have the geographical repartition of different subspecies of Morpho-Eleanor throughout South America, and you can see that there is large divergence in color pattern between all of those morphos found in different locations. And what is also interesting is that in some locations, Morpho-Eleanor lives in sympatry with a close sister species called Morpho-Achilles. So they live in the same environment, and you can see that when Morpho-Eleanor and Morpho-Achilles lives together, there is a strong convergence of the wings of their color pattern in those localities. So those butterflies live in the same environment, they are very similar, and despite those facts, we found no gene flows between those 2 species. So we wonder whether some pre-mating barriers could exist between those 2 morphobutterflies, and whether visual cues could be used as pre-mating barrier cues between those 2 species. So in order to answer this broad question, I have divided my talk into 2 different parts. First, I'm going to study whether visual cues are used in mate recognition in Morpho-Eleanor in particular by comparing the differences of coloration, visual perception, and behavior of two Morpho-Eleanor subspecies living in allopatry, Morpho-Eleanor bristowi and Morpho-Eleanor theodorus. And second, I'm going to test whether those visual cues used in species recognition between the sympatric Morpho-Eleanor and Morpho-Achilles I just presented before. So I've divided my method into three parts. So first, I have measured this very complex and changing phenotype that is iridescent by measuring the reflectance of each of my morphos at different angles of illumination. And this means that for each of my morpho individual, I had a multivariate data set to analyze in order to quantify their iridescent coloration. Second, I've used visual modeling and used the already published sensitivity data of the morpho-visual system, Morpho-Eleanor visual system in the literature to build a mathematical visual model in order to test whether the differences of iridescence are found in the color quantification could be perceived by their visual system. And finally, I have performed behavioral experiments in order to test whether in real life, in real conditions, morpho use visual cues during courtship. So I did, for this, I performed male choice experiments based on visual cues only. So the males have had the choice to choose between their own conspecific female or another morpho female. And I've built female model with real female morpho wings that I washed with hexane in order to wash them from any other pheromones and only test the use of visual cues during courtship. So for the first part, our visual cue used in mate choice in Morpho-Eleanor. Here I'm going to present a very sum up data of the differences of coloration and iridescence, so the variation of coloration at different angle of illumination are found on my morpho wings. So on this PCA, each point represents the iridescent global signal of one individual. And the closer the points are together, the closer the iridescent signal of the individual is. So in purple, you can see the iridescent signals of Morpho-Eleanor bristowi individuals, in dark purple, the males, and in light purple, the females. And in orange, this time, it's the differences of iridescent signals of Morpho-Eleanor theodorus, sorry. And in males the males are in dark orange and the females in light orange. And from this graph, both visually and statistically, we notice that there are significant differences of iridescent signals between the males and the females of those two subspecies of Morpho-Eleanor, but also significant differences of coloration between those two subspecies. So they belong to the same species, but have different variations of blue coloration on their wings. Now the question is, can those differences be perceived by a theoretical Morpho-Eleanor vision model? So here is the results of my visual modeling. So on this graph, you can see on the y-axis the chromatic distances of measured at every angle of reflectance I measured on the wings of my morphos. And the chromatic distances can be used as a proxy for discrimination. So the higher the chromatic distances are, the better the differences of coloration can be discriminated by a Morpho-Eleanor visual model. And you can see the dotted line is the threshold of discrimination. So if the points are higher than 1, it means that the coloration can be perceived by my Morpho-Eleanor visual model. And here you can see that at least some chromatic differences can be perceived by the visual system of Morpho-Eleanor between those two subspecies. And my next question was to test whether this is the case or not and whether they use these visual discrimination capacities during courtship. So here is the results of my mate choice experiment. On the x-axis here, I'm going to show you the probability for my Morpho-Eleanor theodorus males and Morpho-Eleanor bristowi males to approach their own consubspecific female. And you can see here that Morpho-Eleanor bristowi males, only using visual cues, tend to visit more often their conspecific female. But there are differences of coloration and iridescence between Morpho-Eleanor theodorus and Morpho-Eleanor bristowi, but you can also see that those two butterflies have different wing pattern, and Morpho theodorus has a smaller band on its wing than Morpho-Eleanor Bristowi. So I wanted to test whether pattern detection or color recognition was involved in mate choice. So I put my Morpho-Eleanor Bristowi males in a cage, and I gave them a choice, mate choice, to choose between a wild-type Morpho-Bristowi female and a Morpho-Bristowi female modified in order to resemble the pattern present on the wings of Morpho-Eleanor Theodorus. And I found that the Morpho-Eleanor Bristowi males tend to visit significantly more often their wild-type females, meaning that maybe pattern detection could be involved in mate choice in Morpho butterflies. So on this part, are visual cues used in the Morpho butterflies in mate detection in Morpho-Eleanor? Yes, Morpho-Eleanor can perceive visual cues during courtship and mostly pattern differences. And so this begs the question, what happens when Morpho-Eleanor lives in sympatry with another Morpho species, and what happens when both of these species have the same pattern? So I did the exact same experiment, but this time I compared the coloration, the discrimination capacities, and the preferences during courtship between Morpho-Eleanor and Morpho-Achilles living in sympatry. So this time, this is the same type of graph than earlier. Each point represents the global signal of iridescence between those two species. And what you can see here, and the main difference compared to before, is that this time there is still significant differences of iridescence between males and females, but between those two species, there are no significant differences of iridescence between those two species, meaning that their blue iridescent coloration is very similar. And in fact, when I analyzed these differences of coloration with a morpho-visual model, I've found that the chromatic distances are always found below the threshold of discrimination, meaning that those differences of coloration, in theory, cannot be perceived by a Morpho-Eleanor visual model. And in fact, when I performed mate choice experiments, I found that neither the Morpho-Eleanor males nor the Morpho-Achilles males prefer their conspecific female, and they just roam in the cage and visit both female equally. So no color discrimination of their conspecific mates. So here is a global conclusion. So yes, Morpho-Eleanor and Morpho can perceive visual cues during courtship and mostly pattern differences, but those visual cues cannot be used as species recognition cues between Morpho-Eleanor and Morpho-Achilles in sympatry. So maybe the visual discrimination is not involved in species recognition in this particular case of sympatry in Morpho butterflies. And for those who study butterflies, you know that there are other cues that can be used as species recognition in butterflies, especially olfactive discrimination. So just as a quick perspective, I wanted to tell you that right now I'm studying the differences of molecular compounds found on the genitalia of Morpho-Eleanor and Morpho-Achilles living in sympatry. And so far the results show that there are significant differences of molecular compounds found on their genitalia, and this kind of patterns, the differences of potentially pheromone between males, very significant, could be the sign of an olfactive discrimination by female and female choice in this case of sympatry. So I would like to thank you for your attention. I would also like to thank my supervisors and my collaborators, and I would be happy to answer some questions if I have some time left. Thank you very much. Yes.
So when you showed the visual sensitivity of these butterflies, you showed that they have two opsins that are sensitive to blue and two opsins sensitive to green.
Yeah.
Did that happen through duplication, you know? And do you know what could they be used for? Because it seems that they're quite close, so there could be some color discrimination.
Yes. So apart from this project, I've also been studying the evolution of the visual system of morphos at a larger scale, at the family scale. And those differences of sensitivities can either be due to duplication or to filtering pigment that could exist on the eyes of morphos, but so far we have no idea which is it. Yes?
Did you look at behaviour at all? And I mean, was behavioural information available in the make-choice trials for the wing stunting?
So behavior in terms of like.
Because obviously the way that they move the angle of the wing significantly influences the temporal dynamics of the information coming into the eye.
So I have not quantified this, but I know that, for instance, I've compared the mating behaviors of Morpholinor bristoi and Theodorus, and I noticed that Morpholinor bristoi tend to flicker their wings like this, and Morpholinor Theodorus do not have this behavior at all. So there is maybe an effect of different also mating behaviors between those two Morpholinor subspecies, but I have not properly quantified this. Thank you very much.
Thank you so much for attending this symposium. I have been so impressed with the talks that we have been exposed to so far. So thank you to the organizers. Today, I would like to start this by thanking three individuals, Patrice, Brett, and Luke, who have been the ones that collected most of the information that I'm going to share with you today, along with some other collaborators and, of course, the funding agencies. In the last few years, we have seen an increase in the number of papers related to the comparative aspects on avian vision. And this has been super exciting. Most of the main reason is because decades ago, whoever was studying comparative avian vision would not necessarily control for the effects of shared phylogenetic history, right? But these papers are doing that, and they're doing an excellent job. Most of these studies try to look at the relationship between anatomical or physiological traits and some kind of component. It could be an ecological component, it could be an environmental component. And they run tests, right? Again, controlling for phylogenetic effects, and the tests tell us whether there are significant differences or not significant differences. And we should keep doing the same, that's great. But today, I wanted to see if we can start thinking and adding another component, which is the component of effect sizes in these anatomical or physiological comparisons. So in this context, the way I'm defining effect size is the degree of difference between two species on a given trait. So here we have an example. We have trait A and trait B, right? We have two species. We could have many more, of course. And on trait A, we can see that the difference between the two species is small, and that we call that a small effect size. But in the lower portion, we see that the differences between the two species on trait B is much larger, and we call that a large effect size. I guess the question that I would like to bring up is, what does the degree of differences in these traits mean for visual performance, right? Obviously, with this talk, I won't be able to answer that particular question, but I'll try to attempt to begin the process. So, what we decided to do is to look at the effect sizes in visual traits between two groups of birds. Species that detect food at close distances and species that detect food at far distances. And, you know, we can loosely call the species that detect food at far distances predators, and those that detect food at close distances prey, although it's not necessarily appropriate for obvious reasons. And we are focusing on terrestrial diurnal species that locate food mostly using the visual system. So the traits that I'm going to be looking at are related to spatial vision, related to the visual field configuration, and also related to visual acuity, right? So we have the sphere, and the bird is at the center of that sphere, and where the bill projects, in that gray area, we have the binocular field, right? The lateral fields on each side of the head, and then on top of the head and towards the back, we have the blind area. And you also see that red dot that represents the projection of the center of acute vision, which is usually the retinal specialization that Emily was talking about earlier. So we can cut that sphere in half and then take a sort of top view of that sphere, and you can see these landmarks again: the binocular field, the blind area, the lateral fields, right? The projections of the centers of acute vision. And we can also look at visual coverage, right? Which is the 360 minus the blind area. So let's get started with the first trait, visual acuity, which is the ability to resolve detail in an image. In a recent paper in which we controlled for phylogenetic effects, we found that bird species that detect food at far distances have higher visual acuity than those species that detect food at close distances, which is highly expected. And the differences were significant, and that's great. But again, trying to push the idea of effect sizes and filtering for species that are only terrestrial, here is the way I'm going to be showing the results during this talk using these Gardner-Allman figures, in which to the right side you can see the estimation of the effect size and the bootstrapped confidence intervals. And the idea of the confidence intervals is that they give us a sense of the precision of the estimate of that effect size. Okay, so differences, you can see them. The difference between these two, the effect size, is about 28 cycles per degree. So the question is, what does 28 cycles per degree mean, right? So we can use acuity view, right, and we can portray how much different the viewer will have depending on the species looking or detecting food at close distances versus far distances when the viewer is 25 meters away. So obviously, the species detecting food at close distances have a more difficult time, right? But if the viewer is 75 meters away, then the species detecting food at close distances is really limited in the way or the information it has to define what kind of visual stimulus that is. The other trait is visual coverage. In this case, is on the horizontal plane. Remember that top view of the visual field, right? And I told you that visual coverage is approximately 1 minus the blind area width. So here we look at the estimates, the effect size estimate of blind area width. And what this is telling us is that species that take food at close distances have greater visual coverage, right, because they have a smaller blind area than those that detect food at far distances. And the estimate is about 23 degrees. How much is 23 degrees? What does that mean? Well, that's the dark gray area at the back of the head close to the binocular field. That's the delta between these two groups. And it must have some consequences, right? Because those 22 degrees allow the species that take food at close distances to be more reactive to stimuli around their heads. So that's horizontally. We can also look at this vertically, right? And that's going to be determined by the vertical extent of the binocular field, right? That varies quite a bit in different species. So what we found is species that take food at close distances have a higher vertical extent of their binocular field. And the difference was about 36 degrees. And again, what does 36 degrees mean, right? And you can see the difference, that delta, that effect size in this sort of lateral view of the bird's head. Another important trait that we looked into is the degree of eye movement. So what happens with some bird species, some species can move their eyes quite a bit, other species not so much. And it is an important trait because animals can converge their eyes, and by converging their eyes, they increase the size of the binocular field. But they can also diverge their eyes, and in that way, they reduce the size of the blind area, increasing visual coverage. So what we found is that species that take food at close distances have a much higher degree of eye movement than those that take food at far distances. And the difference was estimated to be about 16 degrees. So what does that mean? Well, in humans, we move our eyes on the horizontal plane above 45 degrees. So 16 degrees is about 35% of that. So imagine that we are cut by 35% our ability to move our eyes, right? So that will mean that we need to compensate for that, and maybe we do that by moving our heads, or these animals can do that by moving our heads. So the final trait that we looked at is the number of centers of acute vision, or the number of retinal specializations per eye. And this was pretty straightforward, right? Because all species that detect food at far distances that we sampled had 2 centers of acute vision per eye. And all species that detected food at close distances had only 1 center of acute vision per eye. So the effect size is 1, but remember this is per eye. So around the visual field, that translates into 4 visual acuity spots for species that take food at far distances and 2 visual acuity spots for species that take food at close distances. Okay, so I went through a bunch of descriptive markers, right, on these spatial vision of these 2 groups of species. How can we wrap this up? Well, what we can do with this information is we can start coming up with some functional/mechanistic hypotheses about how these species are limited by information and how they try to gather that information through different compensatory mechanisms, right? So species that take food at close distances, their frontal spatial vision actually has a large binocular field. And that might be really good to improve the visual discrimination and the detection of food items. Notice something, though, that the centers of acute vision do not fall into the binocular field. And I'll be happy to talk more about that in the Q&A if there is time. Also, these species are limited in terms of visual acuity. They don't have a good ability to resolve stimuli from far away. So having this wide visual coverage vertically and horizontally might allow them to detect a bunch of different stimuli around the head to confirm whether or not it's a predator. Also, they have this large degree of eye movement, which can allow them to quickly shift between sort of a food-centered visual system to a predator-centered detection visual system. So in the species that take food at far distances, I told you they have 4 centers of acute vision, right, 2 per eye. But the interesting thing is that these centers of acute vision seem to be almost equidistant from each other. So what we are hypothesizing is that maybe that's pretty useful for these species to time in space and time the movement of these mobile prey, and they will allow them to approach that prey in a more successful manner in terms of capture. So species that take food at far distances, frontal vision may enhance detection and spatiotemporal tracking of food. The second take-home message is that now we have some effect sizes that we can use as benchmarks to guide us in the interpretation of future comparative work. So if we do some study and we find a difference, let's say in visual acuity between species of 5 cycles per degree, what does that mean? Well, you know, we know at least in this group, important difference is close to 30 degrees, right? So that's sort of the way that we would like to start thinking a little bit. And sort of the frontier, which we're not even close to that, is to try to start thinking about the minimum effect size of interest, which is in this context, the smallest difference between species on a given visual trait that would result in differences in visual performance. And that will involve, like many of the presenters in this talk, anatomical, physiological work, behavioral work, and the integration of these things. And with that, I thank you very much for listening. Yes?
Really cool stuff. Have you been able to delve more into, instead of just 2 categories, within the predators, like their prey type? Are there differences if they're like insectivores versus small mammal eaters or something like that?
That's next, right? We haven't been able to get there, but obviously that's what we want to do. One of the challenges is to get all the multiple visual traits measured on multiple species, right? So you know, databases right now have a visual trait on all these species, then in the second visual trait on a bunch of different species, but they don't overlap much, right? So that's what we're trying to do, and it's really time consuming. Yes?
Are you looking into whether the degree of eye movement is related to the nature of displays, sexual displays, and sort of the degree of movement in displays? Because a lot of prey species are the ones that have more elaborate or more dramatic displays.
No, but thank you so much for that wonderful idea. That's awesome. Yeah, no, we are not looking into that, but yeah, I may contact you, so yes, thank you.
With regard to eye movement, what about the whole head movement as well? So swiveling of that effect?
Right, so that's a great question. So we have other studies looking at the relationship between head movement and eye movement, and there seems to be a correlation between the two. Not a strong correlation, going back to the effect sizes, but there seems to be a correlation. We think that the eye movement story is very complicated, and you know, we just had an idea, right? But it might also be related to food demands. It might also be related to the way they trade off trying to detect food with trying to detect predators, trying to detect conspecifics, right, for social interactions, and so forth and so forth. Well, thank you.